Hi viewers, this is Shomala Chatterjee, currently pursuing my masters in biotechnology from Pondicherry Central University. In the last two lectures, in the first one, I discussed about glycolysis and then we discussed about the linking reaction. Remember the locations, the glycolysis occurs in the cytoplasm and the end product of glycolysis was pyruvate. And I already mentioned in the second video that pyruvate, if it gets oxygen properly, sufficient amount of oxygen, the pyruvate in aerobic condition, it gets converted to acetyl coenzyme A with the help of pyruvate dehydrogenase complex. And this conversion of pyruvate to acetyl coenzyme A, it usually occurs in the mitochondrial matrix. So glycolysis, it occurs in the cytoplasm, whereas the linking reaction, it occurs in the mitochondrial matrix. Now, the fuel, the acetyl coenzyme A, which is now going to serve as the fuel, it has entered the heart of the mitochondria. And here, the complete oxidation of the glucose, it will take place. Okay. So welcome to citric acid cycle, which is also called as TCA cycle or Krebs cycle. बहुत सारे नाम है इसका, ठीक है? So do not get confused. Citric acid cycle is also called as Krebs cycle. इसका एक और नाम है TCA cycle. Do not get confused. Okay. This cycle has three names. Again, I'm saying each name has a reason. TCA cycle. Okay. TCA. What is the full form of TCA? Tricarboxylic acid. TCA stands for tricarboxylic acid. Citric acid cycle. Why it is called as a citric acid cycle? Because the first stable compound that you will get in this particular cycle is the citric acid. So इसीलिए इसको citric acid cycle भी बोलते हैं. And it is also called as a Krebs cycle. Why? Because this particular cycle, it was reported for the first time by Sir Hans Krebs. It was reported by Hans Krebs for the very first time. तीनों नाम interchangeably use हो सकते हैं, so do not get confused. Citric acid cycle, why it is called as citric acid cycle? Because the first stable product that you get is citric acid. Why it is called as a TCA cycle? The TCA stands for tricarboxylic acid cycle. ठीक है? और क्रेब साइकिल क्यों बोलते हैं इसको? क्योंकि it was discovered or it was reported for the first time by Sir Hans Krebs. Remember, all the three names can be interchangeably used. Do not get confused. ठीक है? Now, TCA cycle, also called as a Krebs cycle, also called as a citric acid cycle. कहाँ होता है? Mitochondrial matrix में होता है. So the glycolysis, it occurs in the cytoplasm. Be very specific about the locations. ठीक है? ग्लाइकोलिसिस कहाँ होता है? साइटोप्लाज्म में होता है. फिर लिंकिंग रिएक्शन, जहां पर पाइरुवेट को हम एसिटाइल कोएंजाइम में कन्वर्ट करते हैं, वो कहाँ होता है? दैट हैपेंस इन द माइटोकॉन्ड्रियल मैट्रिक्स. एंड व्हाट द नेक्स्ट स्टेप इज टीसीए साइकिल. वो भी माइटोकॉन्ड्रियल मैट्रिक्स में होता है. नाउ दिस पाथवे is also called as amphibolic pathway. What is amphibolic? Amphibolic means ये catabolic भी है, ये catabolic भी है और ये anabolic भी है. ये anabolic भी है. आजकल वेब सीरीज में या फिर सिनेमा में हम विलेन को भी थोड़ा सा ह्यूमन दिखाते हैं, ठीक है? क्योंकि हर इंसान में कुछ अच्छे होता है, कुछ बुरे होता है. सो ये वाला जो पाथवे है, दिस इज एम्फीबॉलिक पाथवे. ये कैटाबॉलिज्म भी करते हैं, ये एनाबॉलिज्म भी करते हैं. दैट मीन्स ये चीजों को डिस्ट्रॉय भी करते हैं, चीजों को बनाते भी है. तो इसीलिए इसको हम एम्फीबॉलिक पाथवे कहते हैं. ठीक है? व्हाई इट इज कैटाबॉलिक पाथवे? क्योंकि अगर हम रेशियो देखेंगे ना, एटीपी और एडीपी का जो रेशियो है, ये बहुत लो होता है. और ये लो तभी होता है जब तुम कुछ चीजों को तोड़ रहे हो. ठीक है? एनएडीएच और एनएडी का भी थोड़ा सा लो रहता है और ये बनाते भी हैं. एटीपी एडीपी रेशियो कभी कभी हाई चले जाते हैं कुछ कुछ स्टेप्स में, या फिर एनएडीएच एनएडी रेशियो भी हाई रहते हैं कुछ स्टेप्स में. सो दिस इज बोथ कैटाबॉलिक एज वेल एज एनाबॉलिक प्रोसेस. इसको हम कहते हैं एम्फीबॉलिक पाथवे. ये चीजों को बनाते भी है और ये चीजों को तोड़ते भी है, ठीक है। नाउ नाउ वी एंटर इनटू द साइकिल, ठीक है। विदाउट वेस्टिंग मच टाइम, लेट मी एंटर इनटू द साइकिल, एंड एट द एंड ऑफ दिस पर्टिकुलर साइकिल I will also give you a mnemonic, okay, so that you remember the cycle and every product that is associated with this particular thing. Okay, so the direct input of TCA cycle is what? Acetyl coenzyme A. Acetyl coenzyme A कहाँ प्रोड्यूस होता है? माइटोकॉन्ड्रियल मैट्रिक्स में। और TCA cycle कहाँ होता है? माइटोकॉन्ड्रियल मैट्रिक्स में। तो acetyl coenzyme A, which is the product of the linking reaction, acetyl coenzyme A, which is the product of the linking reaction, that is actually acetyl coenzyme A, which is the product of the linking reaction, that is actually the first product to enter, that is the direct input of the TCA cycle. Acetyl coenzyme A, remember, it is a 2 carbon compound. Acetyl coenzyme A, it is a 2 carbon compound. Now acetyl coenzyme A, which is a 2 carbon compound, joins with it joins with what? It joins with oxaloacetate. देखो ऐसा एक एरो है ना, so it joins with oxaloacetate to produce citrate, and citrate is the first stable product which is produced. That is why this cycle is also called as a citric acid cycle, and this conversion occurs with the help of an enzyme called as citrate synthase. This conversion occurs with the help of a reaction called as citrate synthase. Now see, acetyl coenzyme A, as I said, it is a 2 carbon compound. Oxaloacetate is a 4 carbon compound. Okay, oxaloacetate is a 4 carbon compound. Acetyl coenzyme A is a 2 carbon compound. दोनों combine होता है in the presence of this enzyme called as citrate synthase. ये citrate बनाएगा, which is a 6 carbon compound, and this is the first stable product of Krebs cycle. This is also called as a citric acid cycle because the first stable product is citrate. ठीक है, अब ये वाला citrate will convert into isocitrate with the help of an enzyme called as aconitase. With the help of an enzyme called as aconitase. ठीक है, with the help of a enzyme called as aconitase. So let me discuss up to this once again. Oxaloacetate, it is a 4 carbon compound. Acetyl coenzyme A is a 2 carbon compound. Both of them will join and produce citrate, which is a 6 carbon compound, and the enzyme that will be used is the citrate synthase. The enzyme that will be used is citrate synthase. ठीक है, अब citrate किस में कन्वर्ट होगा? आइसोसाइट्रेट में कन्वर्ट होगा with the help of aconitase. With the help of aconitase. ठीक है, यहाँ पर कोई कार्बन लॉस नहीं हो रहा है, कोई कार्बन गेन नहीं हो रहा है, क्योंकि आइसोसाइट्रेट भी एक 6 कार्बन कंपाउंड है। आइसोसाइट्रेट भी एक 6 कार्बन कंपाउंड है। हियर देयर इज नो गेन और लॉस ऑफ कार्बन, ओनली देयर इज अ रीअरेंजमेंट। ठीक है, थोड़ा सा रीअरेंज हो रहा है और कुछ नहीं हो रहा है। ठीक है, आइसोसाइट्रेट विल नाउ चेंज इनटू अल्फा कीटोग्लूटरेट। आइसोसाइट्रेट विल नाउ चेंज इनटू अल्फा कीटोग्लूटरेट। यहां पर दिस इज द फर्स्ट डीकार्बोक्सिलेशन विच इज हैपनिंग। आइसोसाइट्रेट को हम ऑक्सीडाइज करके अल्फा कीटोग्लूटरेट बनाएंगे। दिस इज द फर्स्ट डीकार्बोक्सिलेशन विच इज हैपनिंग। ओके, यहां पर द फर्स्ट CO2 इज रिलीज्ड इन द TCA साइकिल। देखो यहां पर एक डीकार्बोक्सिलेशन, दैट मीन्स पहला CO2 हमें ये वाला स्टेप में मिल रहा है। द फर्स्ट CO2 वी गेट फ्रॉम दिस पर्टिकुलर स्टेप। Isocitrate is converting into alpha ketoglutarate. Okay, isocitrate is converted into alpha ketoglutarate with the help of isocitrate dehydrogenase. This is the first what to say, this is the first decarboxylation which is occurring. The first CO2 is released in the TCA cycle, and the compound which is formed is alpha ketoglutarate, और एक कार्बन यहाँ पर लॉस हो रहा है। So alpha ketoglutarate, this is a 5 carbon compound. Okay, alpha ketoglutarate, it is a 5 carbon compound. Now this alpha ketoglutarate will change into succinyl coenzyme A. This will change into succinyl coenzyme A. This is another decarboxylation. Decarboxylation मतलब क्या? कार्बन डाइऑक्साइड जा रहा है। Isocitrate gets converted to alpha ketoglutarate, which is a 5 carbon compound, और ये वाला जो प्रोसेस है This is catalyzed by isocitrate dehydrogenase. Then alpha ketoglutarate gets converted to succinyl coenzyme A with the help of alpha ketoglutarate dehydrogenase. एंड वन मोर थिंग दैट वी हैव टू रिमेंबर, ये दोनों स्टेप में NADH हमें मिल रहा है। देखो, इधर हमें एक NADH मिला, इधर भी हमें एक NADH मिला। तो अभी तक क्या हुआ? वी गेट टू NADH एंड वी गेट टू कार्बन डाइऑक्साइड। वी गेट टू NADH, we get two carbon dioxide till now. So the first decarboxylation and the second decarboxylation both are occurring. Okay, the first carboxylation, the second decarboxylation, both of them are occurring one by one. And in both these decarboxylation steps, we get two NADH. The first NADH, the second NADH. Now, so till now, we reached up to this. We got succinyl coenzyme A. Succinyl coenzyme A is formed. Succinyl coenzyme A is interestingly a 4-carbon compound. Whenever there will be decarboxylation, one carbon will be reduced. So we started with citrate, which is a 6-carbon compound. Here, in this particular step, there was no gain or loss of carbon. So isocitrate is also a 6-carbon compound. Simple rearrangement was done over here. Then isocitrate will change into alpha ketoglutarate. Isocitrate will change into what? Alpha ketoglutarate, which is a 5-carbon compound. Alpha ketoglutarate will change into succinyl coenzyme A, which is a 4-carbon compound. यहाँ पर एक एक कार्बन जो है वो घट रहा है। क्यों? क्योंकि यहाँ पर डीकार्बोक्सिलेशन हो रहा है, और जहां जहां डीकार्बोक्सिलेशन होता है, यहाँ पर एक डीकार्बोक्सिलेशन हुआ, इधर भी एक डीकार्बोक्सिलेशन हुआ, उधर हमें एक NADH मिल रहा है, राइट? Now the succinyl coenzyme A will now change into succinyl coenzyme A will now change into succinate. Succinyl coenzyme A is now going to change into succinate. We can also call it as succinic acid. We can also call it as succinic acid. और यहाँ पर क्या हो रहा है? GDP is getting converted to GTP. ठीक है, GDP is getting converted to GTP. So this is a substrate level phosphorylation, or we can say that ADP is getting converted to ATP. This is a substrate level phosphorylation. This is the only step in TCA cycle where ATP is formed directly. यहाँ पर हमें directly ATP मिल रहा है. We get a direct ATP over here. ठीक है, this is the only step in the citric acid cycle where we get ATP. Now the succinate will change into fumarate. The succinate will change into fumarate, and here we get FADH. So let me highlight, here we are getting one ATP. Okay, हमें एक ATP मिल रहा है, यहाँ पर एक और FADH मिल रहा है यहाँ पर, ठीक है, we get one FADH2 over here, ठीक है, so we get one FADH2 in this particular step, and the enzyme also you remember, succinic dehydrogenase. The fumarate will now change into malate with the help of fumarase, and here we get water. The malate will change into the final product oxaloacetate with the help of malate dehydrogenase, and here we get one more NADH. We get one more NADH over here. So now here we get one NADH, right? Here we get one more NADH, right? Now you just calculate what are the final product we are getting, what are the things we are getting. So if there is a one turn of citric acid cycle, एक cycle अगर होगा एक turn में in one acetyl coenzyme A, if one acetyl coenzyme A enters, what we are going to get? 1, 2, 3. 3 NADH we are going to get. This is the final product. Okay, 3 NADH we are going to get. Okay, along with 3 NADH, how many FADH2 we are going to get? 1. So we are going to get 1 FADH2. We are going to get 1 FADH2. 3 NADH we are going to get. 3 NADH we are going to get. And we are going to get 1 FADH2. Okay. Here we get 1 ATP directly. Here we get 1 ATP directly. So 1 ATP. And we get 2 carbon dioxide. 2 carbon dioxide we get in the 2 decarboxylation steps. So these are our product. Okay, 3 NADH, 1 FADH2, 1 ATP, and 2 carbon dioxide. Now, this cycle will run twice. Why the cycle will run twice? The cycle will run twice for glucose. for one glucose. Okay, so how many NADH we are going to get? If it runs twice, okay, if it runs twice, how many glucose we are going to get, or how many NADH we are going to get? We are going to get 6 NADH, 2 FADH2, 2 ATP, and 4 carbon dioxide. It will be double, right? It will be double. For glucose, it has to run twice, right? For glucose, it has to run twice. So everything that I wrote, 3 NADH, 1 FADH2, 1 ATP, 2 carbon dioxide, it will be doubled. It will be doubled. No glucose carbon remains. Complete oxidation is achieved over here. Okay, complete oxidation is achieved over here. Is that clear? Now, let me tell you the mnemonic that is going to help you to remember this particular cycle. Remember this particular line. I learned this particular line from a YouTube video, so let me share it with you guys. Can I keep selling? Can I keep selling sweets? Can I keep selling sweets for money? Can I keep selling sweets for money, officer? This is a very simple question. Maybe a sweet vendor is asking to someone, can I keep selling sweets for money, officer? Now, whatever lines I have highlighted, see C for citrate, I for isocitrate. Then what? Can I keep selling sweets for money, officer? Right. So this you have to remember, alpha ketoglutarate. You have to remember alpha ketoglutarate. So then this S is for succinyl coenzyme A, another S, it is for succinate, F for fumarase, M for malate, and O for oxaloacetate. So you just have to remember this alpha ketoglutarate. It comes after the okay, so this K is actually for alpha ketoglutarate. So can I keep selling sweets for money, officer? Citrate, isocitrate, alpha ketoglutarate. Then we have succinyl coenzyme A, succinate, fumarate, malate, and then oxaloacetate. So everything is covered. You remember this particular line, a simple question, can I keep selling sweets for money, officer? And you get all the products. But make sure that you remember all the enzymes properly and what are the final products. Okay, because this will help us to understand the final saga of the final part of this cellular respiration, which is very, very important. That is the electron transport chain, which is sometimes also referred to as the oxidative phosphorylation. So till now, if you have any problem, let me know in the comment section.